I think like by the end of this century, we definitely will solve all of heart attacks that are related to high cholesterol. Like, I don't think my generation will ever, like many people would be dying of stroke. Probably the first generation of aging companies kind of failed. The broad strategy that all of them shared was that we would target hallmarks of aging in age-related diseases. I think that strategy makes really no sense. Right now, I think that our therapeutic modalities are still at this like very rudimentary stage. When it comes to writing complexity at the molecular level, those tools are pretty limited. So what we are developing is a form of new therapeutic modality called epigenetic editing that allows you to programmably rewrite the expression status of multiple genes. Sometimes there's like this question in science that like, why is no one asking this?
Welcome to the Free Radicals podcast, where we interview the scientists and builders working to revolutionize biotech, dramatically extend human lifespan, and bring about a sci-fi future where humanity has full control over biology. I'm your host, Daniel Schur, and my co-host is Eric Dye. Today's guest is Lada Nuzhna, a researcher and founder driven by two questions: how did we get here, and how do we stay? Lada dropped out of college to pursue her biology research as a Thiel Fellow and to co-found Impetus Grants, which funds breakthrough longevity science. Through Impetus, she has allocated over $34 million to fund some of the most ambitious projects and clinical trials in aging. She is now developing a new therapeutic modality to target diseases of aging through her startup, General Control. A quick note for today's interview before we jump in. At one point, we discussed why there are no trillion-dollar biotechs, which was true at the time of recording. However, as of November 21st, 2025, this stopped being true when Eli Lilly's market cap reached $1 trillion. The question remains, though, of why there is just $1 trillion biotech and how to build massively valuable companies in biotech. I hope you enjoy this interview.
I think the only sort of first principles way to think about bio is through evolution. Although I do think people over-apply evolution at times and like try to explain too much through the lens of natural selection. But I do think that like everything in biology only makes sense in the light of evolution, even aging. So like the idea that none of our ancestors lived past a certain age kind of leads to the fact that we only evolved to be like robust bags of meat until we're like 35, and past that age we have like evolutionary shadows that makes a human body deteriorate.
Wouldn't aging actually be one of the few examples of something where evolution is actually not a useful frame, right? In the sense that when we look at aging, we're seeing phenomena that we're not necessarily selected for. And so you're seeing more randomness in the biology.
For sure, past a certain age, sort of like until you reproduce, there is a strong selective pressure for you to be more fit, faster, stronger, healthier. After you reproduce, there is no strong reason for evolution to continue selecting for all of those features. We're, in a way, by design, not evolved to live past 120. I think that's the maximum lifespan that was documented to this day in humans.
There's this concept that I've been mulling over since our last couple of interviews with Jacob Kimmel and others, which is this concept of antagonistic pleiotropy. This idea that evolution and natural selection might create selective pressures for traits and capabilities that are beneficial early in life before reproduction, but are actually deleterious later in life once they've lived past their useful kind of timeline. An example of that might be the inflammation system, the autoimmune kind of continuum of like inflammation and autoimmunity might also stem from the same place that allows us to have a strong immune system that's robust against infection early in life, right? So it's kind of an interesting thought process. Have you thought about like, what do you think about this concept?
I think it's a very useful framework also because the way we're used to, like the way we discover drugs in basically every other area of disease is by looking at genetic targets. And I think aging is by far the only area where that hasn't been like as productive of a process. We have some genetic targets for like cholesterol lowering and lipid related disorders, but overall, like genetic targets as a model of early prevention really doesn't make sense in aging, because if you think about what diseases one develops, like when they're 70, it's diseases like IPF, liver fibrosis, all those things. Like it wouldn't make sense for evolution to prevent those early on, because um, the way to prevent fibrosis is to lower collagen production, like roughly. And like if there is no reason for evolution to give that feature to you early in life, because if evolution selects for reduced collagen production, you would just not develop as a young organism. So I think it carries a huge impact on how we discover drugs.
And so then what are the better targets for aging than genetic targets?
I wish I knew. I'm still struggling with this question a lot. I think there are certain areas of aging biology where genetic target selection makes sense, and we do have like genetic targets found in centenarians, like all the ApoE, FOXO, IL-6. But those are so rare and in between that I don't know if it can be a dominating strategy. Like I suspect that we'd need to get some new ways of modeling this highly dimensional biology with new AI approaches, but I haven't yet seen like a convincing way of doing that. Partially because we haven't collected the right data for this kind of stuff.
What do you think of the virtual cell models that a lot of these companies are building? I think focus primarily on like transcription factor activation and the resulting genomic sequencing.
Yeah, I actually I do think perturbation models of a cell are more useful than just collecting the transcripts in longitudinal ways, the way many academic efforts do. So I think in that sense, it's super interesting. I do think our tools are still limited to this day. Um, I mean, if you look at single cell RNA-seq, it captures something like 30% of transcripts within the cell, which is kind of crazy if you think about it. We only measure 30% of the cell state at any given moment. Um, Now, one can argue that you can extrapolate from that using advanced AI models, and maybe it's the case, but I haven't. In a way, I think it would be easier to model a cell than it is to model aging, um, because aging doesn't really happen just at cellular level. It happens at the layer above on the tissue level and also on our organ level.
Something that's interesting to me is this framework that comes from a top-down view of biology, which is if you can define specific functional phenotypes that you're interested in measuring changes around, can you actually do away with more traditional frameworks of molecular biology, which is caring less about am I measuring certain transcripts, are certain pathways being activated, but rather viewing like things in terms of functional outcomes. Like, is this how you view things? Like, what do you think about this framework of like functional top-down biology?
Yeah, I mean, I think the reason people go for a granular readout of a cell is because that's sort of like the layer of biology that has faster response time, and that like to prove something like function or mortality or lifespan, you have to run like a really long clinical trial to read it out and like to power it really, really strongly, and that would be like super expensive endeavor to undertake. And with something like RNA-seq or say LDL, if your treatment is working, you can almost immediately read it out in a blood. Um, I do think that there is no other way for us to run a late stage trial, like an outcomes trial for aging, without being able to measure all those metrics that you mentioned. Like, I mean, right now it would be something like frailty or 6-minute walk test or like your overall function as a person. But those, like, the reason people don't do it more often is because it's like such a high bar to improve that.
Tell us about the Impetus Grants. You led Impetus Grants as someone just recently dropped out of college, right?
Yeah, yeah. I mean, I dropped out of college to run Impetus in the first place. So Impetus was this experiment in funding science that I helped launch around 2021. The idea was that a lot of the science funding that happens in academia is fairly incremental. Often, when you're submitting a new grant or trying to do new research, you essentially have to have your proposal partially completed to get any funding. There are like many historical examples. One that I always think about is Kathleen Carrico, who was obviously recently got a Nobel Prize for mRNA vaccines, but back in 2005, earlier she got demoted from her university when she couldn't get funding for that type of work because it was just like too out there. And I think it's especially sort of significant in fields like aging, partially because the field is just so new. We have no fundamentals. Like, we don't know what aging is. We don't have biomarkers of aging. We don't have good models of aging. And like, if you're funding incremental research at this stage of exploration, basically are not getting anywhere with it. And so Impetus was really the idea of putting being the first check into crazy ideas that would have been otherwise rejected by other funders or somehow didn't fit within existing system and infrastructure. So like, wouldn't be funded by VCs, wouldn't be funded by government, like can be pursued for free. And there are a lot of ideas in that sort of great space in between.
What was the original impetus for Impetus Grants? Was it a set of conversations you had with a specific individual, or were you going into a pre-existing initiative? How did you get involved?
I mean, I think the observation that we had back then was the launch of fast grants. Like, everyone was really frustrated with how the funding for COVID was going. We had no trials very early on. No one was developing anything. It was it was like really painful to change the direction of existing grants to go and work on COVID-related research. And so fast grants launched a few months before that, and they allocated millions of dollars for vaccine development, for early trials, which government couldn't do on quite the same time scale. And I think that was sort of like the impetus for Impetus. We realized that it's actually not just COVID, it's the rest of science funding looks like that. And it makes sense to extend that kind of initiative. It was independent of fast grants, but they were definitely like a big inspiration.
As you look at the scientific funding landscape now, my sense is that things have actually gotten harder in the past few years since the conception of Impetus Grants, right? We look at the NSF and the NIH and many of the non-for-profit government sources of financing for life sciences. They've actually gotten harder and harder to get access. Like, what is the path forward for biotech and for academic life science research?
Yeah, I mean, for better or worse, it's academic funding for science has gone through a lot of change right now. I think the change has been long overdue, and it's probably a painful process for people inside the academia, but a lot of things that were being funded shouldn't have been funded. A lot of things that should be funded weren't being funded. And I think in a way, a lot of science that happens today happens through forms of patronage, the way it used to be hundreds of years ago. I think many of the labs that we see today are really surviving by building relationships with science funders personally and directly. That's sort of like the top labs that you see in academia, like able to do that.
If you were put in charge of the NIH, what would you do?
Yeah, I mean, I think first restructuring NIA would be a big thing. Like, NIA is today sort of National Institute of Aging. You would think that the things that they're funding are actually related to aging, but actually like more than 60% of it is Alzheimer's research, which is just like one little aspect of aging. And we now know that a lot of that research was fabricated by top labs. I don't know if you guys seen that investigation a few years ago by Science magazine called Blots in the Field, where they suddenly discovered that many of the Western blots in Alzheimer's research papers, just like photoshopped the line into into the spot where they wanted to see it. And so the whole beta amyloid hypothesis, which is where majority of NIH funding was going, has like sort of shaky foundations. So I think just redirecting that funding directly for actual aging research would be a change that would have to make happen in the next let's say 10 20 years, hopefully sooner. Apart from Alzheimer's research, a lot of that funding goes towards things like care for elderly and behavioral research of old people, which is maybe valuable topics, but not as valuable as actually curing them or preventing their age-related diseases. I think NIA did fund some very cool initiatives. One of them is Interventional Testing Program, ITP, but those are not getting nearly as much attention as they should be getting.
One of the grand challenges, I think, with solving a problem as large as aging, or many of the other grand scientific challenges ahead for humanity, is that there are obvious deficits with the way that we currently approach most problem solving and most capitalist endeavors, which is that we have this forcing function around myopic behavior, right? If you're a public company, you have to be thinking about, let's say you're a pharmaceutical company, what is the expiration kind of timeline for my most valuable patents, and how do I fill that gap of like loss of exclusivity that I'm facing with my patent cliffs? And so you have like this quarter to quarter, sometimes year to year LOE gap that you're constantly looking to bridge. And so you're constantly forced to go towards these incremental solutions and just slightly better than before drugs in order to fill that gap. And that same kind of myopic behavior is reflected across a lot of different human systems. If you look at academia, it's the same thing. You have this publish or perish kind of paradigm that forces you to do a paper that's incrementally better, but in the same rough general idea space as your prior body of work as a lab in order to continue to get grant financing and so on and so forth, right? So when you think about why that's a challenge, you look at a space like longevity, you need to have completely different frameworks around understanding biology and modulating biology. And these are oftentimes 10, 15, 20-year endeavors that require such a massive leap and such a large upfront commitment to saying we're here for the long run and not for the next year or next quarter. Like, what fundamentally needs to change for us to move from paradigm A of myopia to paradigm B of truly long-dated, long-sighted viewpoints?
Yeah, I feel like new incentive structures is sort of like a big gap right now. Like, if we had an aging drug, say someone went and ran the first aging prevention human clinical trial, next question becomes sort of like, who will fund it? Who will reimburse it? If the drug is still on patent, if it's generic, then no one would run the trial in the first place, and all of those trials would happen in academia. But if it's still on patent and the company can actually profit from approving that drug, will we get private insurers to cover it? Will we get Medicare to cover it? Medicare is the usual answer, but Medicare only kicks in sort of after 65. And so it's really like for anything, like for any intervention that has to go into younger patients, Medicare wouldn't cover it. And then for private insurers, there's really no reason for them to pay for solutions to aging, because the payers that would benefit from those solutions is actually Medicare, which kicks in later. And so I think it's sort of like a broken incentive structure. It doesn't necessarily apply to all the other countries, but it definitely applies to the US. And I think fixing this fixing this multi-payer system, and I don't know what the actual solution is. Maybe you guys have better ideas on this. Would sort of like propel more companies to go and try to develop aging drugs. Because the science is there. We have so many things that we know to extend lifespan in mice. We even have some that extend lifespan in monkeys. We have GLP-1s, which might extend lifespan. And yet no one has ever run the first aging clinical trial.
Do you think of GLP-1s as a longevity drug?
Yeah, I think for me, it's like a question of like, do we believe that caloric restriction extends lifespan? Um, is basically like an equivalent question to ask. And the answer to that is sort of. Well, we run two big monkey studies, using caloric restriction. Then one of them was done by NA, and other one by I think University of Wisconsin. And both of the studies came to different conclusions. One of them was like, yes, calorically restricting monkeys does extend lifespan. Another one was, no, it doesn't. And the difference between those two studies was the control group. So if you put monkeys on the Western diet, um, caloric restriction does have impact relative to the control group. If monkeys just eat very normally, um, the the impact is much smaller or non-existent. And I think given the sort of Western diets that most of us consume daily, I think even like the healthiest people would benefit from some form of caloric restriction. Um, so I do think that if we were to run GLP-1 longevity trial, we would see a pretty big effect size, especially on Western populations.
I never pictured monkeys eating a Western diet, picturing a monkey with like a McDonald's burger.
Yeah. Yeah. Well, in mice, it's interesting because even the normal mice diet that they usually put mice on for studies makes mice chunky. So when they put mice in a Western diet, it's like way out of distribution of normal feeding, which is like a big criticism of many sort of weight loss and cardiometabolic preclinical studies, but that's a different topic.
Lada, how much can you tell us about your startup that you're working on?
Yeah, I expect that by the time this episode is out, we'll be out of stealth. But sort of the big picture and motivation and the observations that I had while running Impetus was that tool development is upstream of many things in biology. If you were to go 30 years back or like 40 years back, actually at this point 50, but like what would be the most impactful thing for you to work on? And it would be developing new sequencing techniques. It would be developing the first ELISAs. It would be in developing the first PCR reaction, because those things really were multipliers of everything else in aging research. Um, and like one big example that always comes to mind is sort of Human Genome Project. It cost us something like $3 billion to sequence the first human genome, and it took us several decades to get there. Today, it takes us a few $100 and a few days to assemble one human genome of a similar scale. And so the question I always had is, should we have started Human Genome Project by developing better sequencing approaches? Because most of Human Genome Project was completed with like shotgun and Sanger sequencing, very little automation, very few computational approaches to assemble the genome. Should we have started there? And so I think something similar is true with aging bio. Right now, I think that our therapeutic modalities are still at this like very rudimentary stage. We have antibodies, small molecules, siRNAs, we sort of have mRNAs. But when it comes to writing complexity at the molecular level, those tools are pretty limited. I mean, the best we can do is like try specific antibodies for 3 targets. But aging is, and especially aging disease, is no one thing. So what we are developing is a form of new therapeutic modality called epigenetic editing that allows you to programmably rewrite the expression status of multiple genes. And I think epigenetics really sort of became big in aging space just in the past decade. We now have sort of natural transcription factors approaches, companies like New Limit and Retro and Altus. What we are developing is a more controlled version of a transcription factor. I call it a synthetic transcription factor, if you will, that allows you to target any gene, any combinations of genes, and durably write their program.
Yeah, this is related to this concept of epigenetic reprogramming, but what you're doing is both similar and different, right? If we look at companies like New Limit, what they're ultimately trying to do is do broad scale epigenetic reprogramming from an older state to a more youthful state, but we're describing as more targeted epigenetic programming. Can you describe the difference for us?
Yeah, if you think about natural transcription factors, they bind more than 1,000 genes at the same time. And while it's a very sort of like hammer-like powerful approach, you don't really have control over which genes those transcription factors bind. What we are looking at is sort of a more granular control, like scalpel-like control of epigenome, where we still can program epigenetics at scale, like say 10 genes at the same time, but we are not, we have like full control over which genes those are. So that's sort of like the main difference between, I would put sort of all epigenetics medicines on the scale where on like one end of the spectrum, we see something like old school HDAC inhibitors, which just reset everything used in cancer, can be quite toxic. And somewhere in the middle would be all the transcription factors that humans have. And the other end of the spectrum is sort of control, precision, writing of epigenome. And I think all of them have their time and place in treating the diseases. But epigenetic editing sort of is like a quite new concept.
And you sort of described it as synthetic transcription factors. Is that the right way for me to really think about the mechanism here? Like, does this look like an RNA therapy that codes for a protein that is a sort of novel transcription factor that does not exist naturally?
Exactly. Yeah. So what transcription factor is in nature fundamentally is a DNA binding domain and some form of a factor that goes and modulates gene expression. And that's exactly what we're developing. There are many forms of DNA binding domains. Sometimes it's zinc fingers, sometimes it's companies like Mammoth and Scribe are developing CRISPR-based DNA binding domains and a form of a factor that is fused to that. But you're correct in saying that it is encoded in mRNA, packaged into a lipid nanoparticle, and delivered into humans. There are many ways of delivering it, but that's sort of the one that we are going with because I think, unlike gene therapy, cell LNPs will really be a big deal in like transition genetic medicines from rare populations to chronic common diseases.
And when I think about transcription factors, and I want to make sure I'm like broadly understanding the space correctly, like I think about how there are transcription factors that can bind to specific sites and promote expression of those sites in the genome. But there's also transcription factors that modulate like methylation, acetylation, which might have, I guess, sort of bigger impacts on whole areas of the genome that are accessible or not. If first of all, is that like framing right? And if so, I guess what you're describing is sort of the more targeted former approach.
Yeah, it can be both, but so we are using different approaches to epigenome writing. It can be methylation, it can be histone acetylation. Sometimes it's a combination of those things. But fundamentally, the fact that we are focusing on is sort of making gene go up or down, because if you can write that, then you can write different combinations of programs. And transcription factors like Yamanaka factors can do that for many areas of a genome and open chromatins in many areas of a genome.
When you're thinking back, Lada, to the origin and inception of this company, what was the framework you used to map the space and then arrive at this particular approach of targeted epigenetic reprogramming as the right space to build?
Yeah, I think it really started with a basic science question. I had no intention of starting the company. In fact, I like actively resisted the idea. I was like, I'll pick whatever best form for contributing to aging is. And it's not always a company. I was working on Impetus, started doing my own research, and was really sort of interested in the basic science question of what's up with methylation clocks. I was just so frustrated that everyone was using them around me, even though we didn't really know whether the clocks are correlative causal to aging, but sort of became this thing that every consumer bio company was doing. It's like telling you your biological age. They're even more popular now. And I was trying to fund a project through Impetus that would go and perturb certain methylation sites that appear in the clocks and see what happens phenotypically. to see if there is any causality, because maybe clocks change downstream of some like fundamental aging process. Maybe they're upstream of it. No one has ever answered that question. And so I really went and talked with a bunch of labs that had tools for writing the methylation status of the clocks. We didn't end up doing the project in the original forms that I suggested, but in the process of talking to those labs, I just realized how cool that technology is. I was like, wow, we really have no other therapeutic modalities that allows you to write cellular programs at the scale. And yeah, I just fell in love with the idea.
What are the big challenges in developing that type of modality?
Yeah, it's a very interesting question. I think a big one is sort of thinking about the first applications of your technology, because often new therapeutic modalities exist on this like shaky ground where they only have one chance to demonstrate the applicability of the technology, and if that one chance fails, the whole field sort of tanks. I mean, like if you look at editors, they were pioneers of CRISPR. Their first clinical program didn't end up succeeding, which set back company by like at least a decade, at least relative to CRISPR, which ended up approving their first CRISPR therapy. So finding the first correct application for the technology is sort of the question that all tool developers should answer, partially because it can be just slightly better than existing modalities. It should fundamentally address a big unmet need, and biology and biotech, I think, has this like better than Beatles problem in that many of their existence or approaches are off patent, much cheaper, have better safety profile, and so that's the bar that your new unproven technology competes against. And so for you to compete with established therapeutic modalities, you should really go and solve like big gap in medicine. And identifying that big and solvable gap in medicine is, yeah, it is, I think, a big challenge for any new therapeutic modality.
And what is the gap that you identified?
I don't know how much we can disclose for this now, but I think two cool things that epigenetic editing can do that few other sort of modalities can do is just rewrite the programs that cell runs. So we are very much used in medicine to going downstream of cellular programs in the cell and just poking those single proteins with siRNAs and small molecules, but epigenetic editing just rewrites the programs that the cell runs. And there are some cool public examples of this that are now in clinical trials, like permanently lowering your cholesterol. Like companies like Tune and Tune and Chroma. They had very cool NHP data showing that you can deliver a genetic editor targeting PCSK9, not edit anything in your genome, and lower your cholesterol for many months without you continuing to take statins or PCSK9 siRNAs like inclisiran.
So we have like CRISPR, which is precision gene editing, and then we have what you're working on, which is precision epigenome editing. Do you see, precision epigenome editing as being more important for aging in the long term than CRISPR, or do you see these as things that are going to work synergistically with each other?
Yeah, I mean, what's interesting about CRISPR and epigenetic editing is that in many cases, they end up competing for the same target space. So now you see many CRISPR companies actually fully pivot to epigenetic editing. I do think that epigenetic editing to aging would probably be similar to what CRISPR was to rare monogenic disorders. But I don't like framing things in like more or less important way, because I think I'm generally open-minded about what would be the solution to aging. I think will likely require multiple. Um, and yeah, open-minded about what other therapeutic modalities people are developing. We funded many of those through Impetus that led to creation of like companies like Radar Therapeutics, sort of radar sensing, early RNA sensor technologies, cell therapies, all of those. I'm excited about all of them.
We talked about methylation clocks earlier. Do you have a theory that you like around like what is going on with methylation clocks? Is that one of the causes? Is that upstream of aging? Is it downstream? What do you think?
I really think we still don't know that, and somehow there is almost no momentum to go and figure it out. There is momentum to use them right and left, and I think even Function Health now incorporates those in your standard blood panel. But yeah, I still think we are at the stage where we don't know what happens with the clocks, and that's very unfortunate because it would be great if we had a blood-based readout of aging, because it would significantly accelerate all of our clinical efforts in running clinical trials for aging.
What's the bottleneck to understanding the relationship there? Because in my mind, we've mapped out a lot of the effects of aging, or at least labs are doing it right now in terms of the like single cell genomics, right? Like you can look at a single cell, and as it ages, we can see that its gene expression changes, right? And we've like correlated that to age. It would seem to me that methylation clocks are like methylation is regulating the genes that are expressed. It seems to me like there's a clear link there. Is there some, I'm sure I'm oversimplifying, right? Like what's the challenge in potentially linking the methylation sites to the sort of aged pathogenic gene expression?
Yeah, I mean, one of the things is like showing that they're causal. So going and directly intervening on those methylation sites and seeing what happens, and coming to answer that question with a very open mind, not trying to find an answer where there aren't any. But a second sort of component to this is showing that methylation clocks can be responsive to aging treatments. Sort of showing that the drugs that we know to extend lifespan can in fact affect the methylation status of the clocks. And that would mean that your sort of biomarker readout is responsive enough for you to go into clinical trials. And I think there were some early efforts trying to see whether methylation clocks do in fact pick up on aging treatments that tend to extend lifespan in mice, but the results were inconsistent. I think there were some methylation clocks that couldn't pick up on rapamycin, and we know that rapamycin is sort of one of the most robust treatments that extends lifespan in mice. So it's definitely tricky. Plus, there are like so many clocks. Yeah, there are so many clocks. And they all sort of have different methylation sites, and might be picking up on different aspects of aging processes.
The way that I think about methylation clocks is that they seem to be really consistent, measurable, assayable kind of systems for understanding the relative age of a cellular, like a cell or a tissue or an organism. But where they really have a lot of struggle in proving causal relationship with aging is, can you identify very specific genes that are consistently methylated with age? Where if you target those genes for demethylation, you can also show there's a causal relationship with longer lifespan or increased function in some important area related to aging. And that's where like the relationship breaks down, because so many genes are methylated. It's very hard to identify which genes are actually, like important for both the clock and for, driving aging forward.
Morgan Levine she's one of the early clocks people who pioneered some of the first papers in the field. She sort of had an interesting paper a few years ago showing that you can construct different clocks from multiple different methylation sites without loss of accuracy, which is fascinating to me. Like, I would expect that, like, say you have thousands methylation sites you're picking from, and you construct like a subset, like clocks from subset of the first 100, and then the clocks of the last 100, and then you compare the two, and they're basically giving you identical results, which is a crazy statistical sort of intuition that I think isn't really making sense for me right now.
Yeah. I guess like the question, the implication there might be that there are some upstream common shared mechanisms of broadly changing methylation profile across the genome that are not genome subsection or loci specific. And so if you looked at any section, it's like for example, of an aging house, like, oh, the timber in this section is similarly deteriorated as the timber in this section, or like the paint chipping in this section is similar to another section. But simply adding another coat of paint on the house isn't going to reverse the fact that it's like an old integrated house, right?
I like that
Um,
analogy. Yeah. Have you done your
So
methylation clock sites? Like, have you measured your methylation age ever?
I have not. Have you, Eric?
It's good. Uh,
never.
yeah. I think it's like not even like the problem with clocks is that it's not even an actionable metric. You sort of get one number back and you're like, well, okay, what do I do now?
I,
Whereas any other readouts tell you like, oh, you have to take more vitamins or you have to lower your LDL. Like, at least you can do something about that.
so I'm just gonna ask a dumb question again, but it's like, all right, we have these methylation sites in a clock, and I can, I know the algorithm, right? So I say, okay, if we undo these sites, you know, the cell will be younger. Like, is anybody taking a cell and just editing those specific methylation sites through like a precision epigenetic, editing tool like what you're working on? Like, is anyone just testing that directly and seeing the result?
So that was exactly the question I was asking, like maybe like the year 2022. I was just going around and was like, why has no one done it? Like, it's like an obvious thing to do. We funded a few projects that try to do something similar, but clocks do have like hundreds of sites, and epigenetic tools are just not yet there to be able to precisely write at that scale. There are some other approaches to doing that that I hopefully those papers will come out in the next few years. But yeah, definitely like a big gap. Sometimes there's like this question in science that like, why is no one asking this?
What are other questions that no one's asking?
Um, yeah. Why, like, I think a big one is like models for aging. Most of the diseases that we have, like IPF, MASH, Alzheimer's, Parkinson's, mice do not develop naturally. And yet, all the treatments that go into human clinical trials for those diseases are tested in mice. And usually it's some form of artificial model of like, we delivered the same as folded protein to the brain of mice, and so that mice suddenly develop dementia. But like basically all the treatments that are being tested in mice fail in clinical trials, and it's true for basically every single age-related disease. We're just so bad at treating all of those. And I think it's very interesting because you can see a clear connection that once we have an animal model of a particular age-related disease, we suddenly have multiple drugs for that age-related disease. I mean, cardiovascular, like LDL lowering is one example of that. I think, like by the end of this century, we we definitely will solve all of heart attacks that are related to high cholesterol. Like, I don't think my generation will ever, like many people would be dying of stroke because we now have PCSK9 gene editors. We have statins. We have inclisiran, all those treatments. I think like John Maraganore recently launched a D2C sort of cardiovascular outcomes prevention company. I don't know if you saw that, guys. Basically, like by the end of this decade, we won't have strokes and heart attacks. And connection I see there is we have more or less good models and biomarkers for that aspect of aging. Um, Finding similarly good models for Alzheimer's, Parkinson's, you name it, would be like a big unmet need to solve. And I don't know what those are. It might be that we have to just test everything in monkeys, which would be very expensive. Maybe there are like alternative animal models, but right now I don't think we're testing age-related disease drugs in correct models.
I do like the idea of testing in more human like in vitro or just preclinical models. I think of people like the team at Bexorg who are, restoring, old, recently deceased brains and basically bringing them back to life so that you can run in vitro studies on this like restored human brain ex vivo. Yeah, so I love these sorts of like really interesting model systems that are more reflective of human biology and clinical outcomes. Another question that came to mind earlier, Lada, was this like really age-old question of like biotech drug discovery, which usually you take one kind of risk on between target risk and modality risk. And in the case of what you're building, it seems like you are pioneering what is still in many ways a novel modality for altering gene expression. But you then have this question of like, where do you point that technology and what targets you're taking on? How did you navigate that question of like selecting the right targets for your technology and balancing risk versus reward for that target selection process?
Yeah, definitely an important question to address, like very early on in company building process. I think that broadly there are two categories of aging companies. One of them is sort of more established modalities, the risk approaches, maybe even already approved drugs that have efficacy and safety in humans in certain indications, and then going after aging as an indication by itself. So sort of taking on regulatory risk. I would even call it maybe a loyal for humans approach, where we would approve the first aging drugs for aging as an indication, which is a big risk to take on. So you don't want to really do novel gene expression approaches there. On the other end of the spectrum, and like one thing to mention about this first bucket is that the effect sizes you'll get from this are likely small-ish. Sort of like if you're working with existing repertoire of drugs, like we're unlikely to have something that fully solves aging there. At best, you can probably get 1 year to human median lifespan, and that would already be pretty good. On the other end of the spectrum is sort of new modalities, riskier approaches, maybe like replacement, where you first go and solve a pretty big unmet need, which would likely be in some form of age-related disease. So you are not taking on any regulatory risk and not trying to develop the first aging drug with like label of aging as an indication. But those approaches long-term would fundamentally give us much larger effect sizes in approaching aging. And I agree with you that sort of like you have to pick your risks, and you can't take on too many. And like with new modalities, it's often both the technology risk and the delivery risk and the indication risk. So you kind of have to remove all those variables and just focus on developing new technology. I don't think one or the other way of building the company is better necessarily. I'm like a big fan of loyal and like big fan of any following loyal for humans approaches. I think both of those sort of have their place to exist.
Lada, you recently wrote an article titled "Where Are All the Trillion-Dollar Biotechs?" which got quite the buzz when you published it. Can you tell us a little bit more about this article?
Yeah, it sort of started with a thought experiment that I had, which is if I wanted to build $1 trillion biotech, is there even a way with an existing infrastructure to do that? Kind of started with the observation that there are several trillion-dollar tech companies, but not a single one in biotech. Yet somehow it's just like pharmas and biotech produce so much value for us as humans. I mean, GLP-1s probably extend median lifespan over the next several decades of the whole population, and yet somehow Eli Lilly is still not $1 trillion pharma. And at the same time, there are more drugs that sell over $1 billion per year than there are software companies doing the same, which is like a very interesting balance. So I just went into like exploring this question with a very open mind. I mean, I think there are like structural reasons for why it's so hard to build $1 trillion biotech, one of which is just like patent life that Eric mentioned earlier in our conversation. There is just like every 5 to 14 years, your value as a company resets to zero, and then you have to build a pipeline that compensates for that. But also just like exploring existing topics within biotech building space that got a lot of attention in the past few decades as like the answers to how to make drug discovery easier, everything from genetic targets to AI to drug repurposing, and trying to answer the question of, can they truly be an answer? Is there a way those approaches would make drug discovery easier?
What were some of the main findings from the article after you ran this research?
I mean, I think there are like broadly two buckets of approaches that we can take on to get to a highly valuable biotech company. One of them is like relative to everywhere else in the industry, can you decrease the cost of developing a drug? And another bucket is increasing sort of outcomes of biotech companies. And there, I think the answer is sort of you have to start going after big indications. Only a few companies at any given point in time pursue age-related diseases. Most of the drugs that we are approving today are not targeting age-related diseases at all. I think there's like 2 or 3 drugs per year in the past few years, with 50 drugs approved yearly, so only a small fraction. I think you have to start going after big indications to be able to get to those like $1 trillion like outcomes. Because if you look at biotech IRR over time, the 2 times in the past 5-10 years when they changed their trajectory was when we had a drug for a big indication. It was COVID vaccines with Pfizer, and another one was obviously GLP-1s.
There is one really interesting section in the article which you titled, "Will AI Help Us Develop the Next Blockbusters?" And help us to understand kind of where AI for drug discovery fits into your framework of understanding the future of biotech, and how important is AI going to be?
Yeah, I had to put a lot of effort into writing that section because I tend to sound overly pessimistic about AI, and I don't mean to be that way. I think the way I would describe my takes on AI drug discovery is sort of short-term pessimist, long-term optimist. And I think where the optimism comes in is like where I think I'll become more optimistic is when we are able to predict efficacy in humans. And yet a lot of the approaches for AI drug discovery don't target that sort of hardest thing. A lot of the companies that are being funded today either go after like cell line or animal model toxicity prediction. And there is still sort of this gap between animal models and human efficacy where a lot of things fail, even if they work perfectly well in mice. I think preclinical work is not where most of money is spent in biotech. Yes, you have to spend like maybe 10, 20, $50 million to develop a drug, but majority of your costs will be in the clinical trials and testing sense in humans, and we don't yet have amount of human efficacy data that would allow us to train the models that can predict efficacy in humans well. I think it will take us quite some time to get there, like at least a few decades.
This topic also seems related to another issue that I think a lot of pharma companies, or at least biotech startups, face, which is that it's very hard, it's exceedingly rare, right, to build a successful platform company, right? There's very few companies that successfully license software to pharma companies to help them with discovery, because ultimately my understanding is your platform's not worth anything unless you develop a drug, right? Like all that matters is the drug. And that makes me think, right, that these pharma companies also, like they don't really have proprietary platforms. What they have is they're really good at commercialization and the regulatory process, and they have a lot of capital. Do you see like the lack of platform biotechs being ubiquitous? Do you see that continuing, or do you think technologies like gene editing or epigenome editing, like, are those things that could enable successful platforms?
Yeah, platform like takes on platforms from VCs and biotech founders is like a swinging pendulum. Every maybe like 5 to 10 years, the field resets their takes on whether platforms are truly valuable or not. I think right now where the pendulum is everyone is pessimistic about the platforms. Everyone thinks, well, if you as a company are only valuable when you have a drug in the clinic, that's where all the exits happen. Then just focus on developing that one asset. No one cares about your fancy technology. I think maybe 10 years ago, no one would have funded a CRISPR company that said that we are not developing CRISPR, we're just developing this one drug and we'll ignore everything else, which is interesting because I think one of the biggest CRISPR outcomes in the past 10 years came from the company that just refused to develop technology by themselves, which is like Verve Therapeutics. They just went and out-licensed their technology from Beam, spent no time in development, paid like quite a licensing fee for that, but was in the clinic much faster than any other company would be in a similar situation. So I think some technologies are more mendable to be in the platforms than others. I think epigenetic editing definitely is, even though I like saying that our platforms will be derived from our products. So I'm still very much like product minded. I think other therapeutic modalities like small molecules much harder to develop into platform because fundamentally you just start with a new backbone each time. It's very hard to engineer small molecules and make them modular in nature in the same way that biologics are.
I think of platforms versus programs, this debate, as being highly reflective of the macroeconomic state that you're currently in. in like eras where you have much more flush capital, interest rates are low, investors have more capital and more enthusiasm and exuberance of deploying that capital, and vice versa on the founder side. I think in those cases, the kind of arms race of the size of outcome that you have to project in order to raise a round and raise increasingly large rounds suddenly gets larger and larger. And so that's the era of like bubble dynamics and bubble economies that cyclically turn back into, program focused, more like greedy search oriented economies where you're looking to just fill up and plug revenue holes that are always cropping up as a pharma company. And right now we have been in a protracted program focused era of biotech, and it's not clear if and when we'll return to the platform era. But interestingly, the one subsection of biotech which has resisted program focus is the AI and bio kind of intersection, right? We look at some of the recent financings from New Limit, from Inveta, from other companies at that intersection. Why is it, Lada, that you think there's been this resistance to the broader program focus of the rest of the biotech economy in this intersection of AI and bio?
I would actually say that New Limit is fairly product focused. I think if you talk to Jacob, he's like very much believes that they have to push actual drugs into the clinic. Their first program is sort of liver failure, alcoholic related liver failure. Um, and but many other companies in the space are doing indeed fall in that category of like, um, let's wait to develop the drug. I think because once you pull a trigger on developing a drug, you immediately start burning more money and you commit to something. And it's much easier to exist in this like exploration stage where you sort of like non-committal, maybe you do a few partnerships, exploring your technology. And you never have to go into the like very expensive part of company building.
Lada, we'd like to end the interview by doing some lightning round questions. So we're going to fire off a bunch of questions and you can give 1 or 2 sentence short answers.
Yeah, sure.
So the first question, a very easy one. What age do you think we'll live to and why?
Um, Psychologically, I don't want to contemplate anything less than however much I want to live. I think there is a worst case scenario and best case scenario. Worst case scenario would probably extend lifespan by maybe like 5 to 10 years, which is like with all the existing cardiometabolic drugs, and we won't be the generation to die of heart attacks. Best case scenario is like an unknown.
Do you believe in the concept of longevity escape velocity?
Um, I definitely do, and I think when we'll get there, like really, like the question is like when we'll get there, and that can be next 50 years, it may be the next 500 years. I think it definitely makes sense. That's at least how I operate. And like we'll start with simpler solutions. We'll solve heart attacks. We'll extend lifespan by a bit, but who knows what will be happening in the year 2100, like what kinds of technologies will exist there. Like I hopefully hope to live to that time to be able to benefit from future medicine.
What percent chance do you give to achieving longevity escape velocity in your lifetime?
I think whatever number I gave it would probably be a meaningless number. Things are changing all the time, but certainly less than 50%.
And how much day to day does the answer you would give to that vary?
I actually don't ask myself that question that often. I know that the number is not that high. I'm definitely not in the bucket of people who believe that aging will definitely be solved. I think some people believe that we already solved it. We just need to push things into the clinic. Definitely more than like cautiously optimistic, but the number is definitely always less than 50%. So we have to get more people into aging space.
Makes sense. Who do you think is doing the most interesting or important work in longevity?
I love when people take on disciplined type of risk. I'm a big fan of obviously New Limit. I think in academia, Vadim Gladyshev has some very interesting pioneering work on ground zero of longevity, which is also concerns the epigenetic state. Um, yeah.
On the flip side, who do you think is doing the most damage to longevity?
Yeah, I think people who sell consumer mutilation clothes. Just because I wish when people first learned about agent space, they learned about concepts that are way more useful than that. And now everyone is sort of used to throwing around the biological age number. Like you'll see the titles every day. We solve biological aging, whereas like biological aging is just like some tiny molecular readout, which we don't even know what it means.
For those who are interested in going into the longevity space, where do you recommend that they get started?
Um, I mean, there are like three buckets: go build something in industry, do in academia, or do something in government. And I think government is sort of this underappreciated direction that few ambitious people take on. I think a lot of the bottlenecks that we have right now are regulatory, and I do hope more smart people go into that direction and work with restructuring NA and also just making it easier for all of us to run first agent clinical trials.
Can you share a contrarian opinion that you have on aging and longevity?
Uh, Let me think. Um, I think uh, broadly, the first generation of aging companies kind of failed. And the broad strategies that all of them shared was that we would target hallmarks of aging in age-related diseases. I think that strategy makes really no sense. I think hallmarks of aging is not really an actionable framework. What hallmarks of aging are is this sort of list of types of damage that happens within the cell, and many of the companies targeted those types of damage, like say senescence and targeting senescence in osteoarthritis. And basically all of those clinical trials failed to this day. I think it really doesn't make sense to target mechanisms of aging in age-related diseases. Yeah.
Do you have a personal routine for longevity that extends beyond sleep, diet, and exercise? And if so, can you share any details?
Um, Not really. I do intermittent fasting. I've been doing it for quite some time now. I think I'm like very much used to it, but it's because it's like a low maintenance kind of strategy. I'm definitely no Brian Johnson.
AI applied to biology, overhyped or underhyped?
Um, Short term overhyped, long term underhyped.
If you weren't building your current company, what longevity biotech would you build?
Um, I think the idea of loyal for humans is long overdue. I mean, one of our shared friends, Alex Colville, is now working on the company like that. There's still stealth, but someone running the first agent clinical trials um, is the idea I would have been working on otherwise.
Lada, thank you for joining us for today's conversation.
Thank you, guys.
Thank you for listening to this episode of the Free Radicals podcast. If you enjoyed this episode and would like to support us, the most helpful thing you can do is to share this with a friend that you think might enjoy it too. And please also leave us a 5-star review on Spotify, Apple Podcasts, and like and subscribe on YouTube. It would really mean a lot. I'm Daniel Schur, and my co-host is Eric Dye. Thanks for listening.